Welcome to the sixth webinar in our Introduction to the Regulatory World. My name is Harald Enzmann.
And my name is Sabine Mayhofer, and we are here at the Bee Farm in Bonn. And from Lisbon, we have Bruno Schepoich.
Hello, good morning. From Lisbon, I'm Bruno Schepoich.
In this webinar, we will look at a number of real cases with CHMP decisions that will illustrate the principles we covered in previous webinars.
First, however, we need to cover the formalities. Harald and I, we are from the German Regulatory Authority, the Federal Institute for Drugs and Medical Devices. Bruno Schepoich is with the Infarmed, the corresponding Portuguese regulatory authority. We all work on centralized procedures with EMA.
And therefore, we have to make this disclaimer. This presentation reflects our personal views. If it is inaccurate or you don't like it, blame nobody but us. In this presentation, we will look at examples for the assessment of efficacy. What is needed to conclude an efficacy? What is needed to come to a positive conclusion on efficacy? We will then look at the pathway leading to a conditional marketing authorization. This is important as this regulatory pathway, the conditional marketing authorization, is used for the most promising, for the potentially game-changing new products. And finally, we will see how we use subgroup results, always a tricking issue, but becoming even more important since our colleagues at the FDA are putting more emphasis now on inclusion and diversity in clinical trials. For each of the topics, we will follow the same principle. Setting the scene and then looking at examples for positive and negative decisions. For the assessment of efficacy, we will have examples how positive decisions were made and examples that were insufficient to justify the positive conclusion of efficacy. For the conditional marketing authorization, we will outline the particularities of this particular regulatory pathway. We will look at positive examples where the conditional marketing authorization in the end resulted in a full marketing authorization, and we will look at negative examples where in the end the marketing authorization had to be terminated. And we will try to give some orientation for future decisions based on consistency with the past decisions. When we come to subgroups, we will distinguish between subgroups based on general criteria and subgroups defined by the presence or the absence of the pharmacological target. Again, we will look at positive and negative examples. With that, let's go into the first big topic, the assessment of efficacy. And for that, over to Bruno.
Thank you, Harald. So let's start to set the scene and setting the scene very clearly, actually. So one thing we all agree on is that although safety may be relative, efficacy is not, and needs to be clearly demonstrated as a fundamental part of the benefit-risk balance of a medicinal product. So the evaluation of the benefit-risk balance is a key element across the entire life cycle of a medicinal product. Benefit risks assessments support decision-making purposes during the development and the regulatory process of marketing authorization, as well as post-marketing follow-up, and also it's useful for the downstream stakeholders such as HTAs, payers, healthcare provider selection. The benefits consider the favorable effects, but also the uncertainties and limitations surrounding these favorable effects. Similarly, the risks related to the use of the medicinal product consider any risk relating to the quality, the safety, or efficacy of the medicinal product as regards patients' health, public health, and effects on the environment. The unfavorable effects are also balanced with the uncertainty and limitations about these risks and how these may be managed. However, acknowledging that the risks related to the use of the medicinal product consider any risk relating to the quality, safety, or efficacy of the medicinal product, the pharmaceutical quality of an innovative medicine has hardly ever been prohibitive of a positive benefit-risk balance, especially when pharmaceutical quality may be improved and the issues identified may be resolved. Safety is always judged in relation to the benefits expected with the use of the medicinal product. There is no independent conclusion on safety, and when assessing safety, the expected benefits need to be considered in parallel, and also how the risks may be managed. Efficacy, on the other hand, is conditio sine qua non for approval. It goes without saying that without demonstration of efficacy, neither an excellent safety profile nor immaculate pharmaceutical quality will be sufficient for marketing authorization. So let's consider again the efficacy in terms of efficacy of medicinal products. Where does this evidence come from? The cornerstone remains randomized clinical trials. For the standard, these are the standard for providing confirmatory evidence on the efficacy of a new treatment. Their prospective, randomized, and double-blind nature provide evidentiary support to the decision-making process. However, we have been seeing increasing exceptions. In a relevant proportion of marketing authorization applications submitted to the CHMP, the pivotal clinical data stems from single-arm trials, the SATs, we will refer ahead in this presentation. And in other situations, more real-world evidence is being submitted for assessment during the marketing authorization application to support evidence of efficacy. This is observed across different therapeutic areas, but also, interestingly, including rare diseases. And I hand back the floor to Harold.
When we now look at the examples for positive efficacy decisions, we will not look at the no-brainers. We will not look at the examples where positive efficacy is so obvious that nobody has any doubt. Rather, we will look at examples that received a positive opinion in the end, but may have been less striking.
The first example will be just that, formally an uncomplicated standard procedure, 2 pivotal trials, both statistically positive for the primary endpoint, supported by the secondary endpoints. Nevertheless, in the end, positive results of the assessment were not that obvious from the very beginning. Gefzuri's gefapixant was developed for the treatment of refractory or unexplained chronic cough in adults.
With other words, this meant to reduce the cough rate. And while we always welcome if innovative medicines use a pharmacological mechanism that is new, the decision will depend on whether there is a clinically relevant effect, meaning, is there a benefit on the coughing that is perceivable for patients? Here you see the results of the primary endpoints of the 2 studies. Coughing, more exactly cough rate, was more reduced with gefapixant than with placebo in both studies. Seemingly simple and straightforward. Be aware, though, that the endpoint is the reduction of coughing against baseline. And importantly, there is a clear improvement in the placebo group as well. The clinical improvement in the placebo group is much bigger than the improvement gefapixant adds on top of the effects seen in the placebo group anyway. We see the same when you look at the secondary endpoints. In both studies, pronounced improvements in the placebo group, with an added benefit in the treatment group. But again, the benefit added by the treatment on top of the improvement seen in the placebo group is much smaller than the improvement seen under placebo anyway. In addition, there have been some issues with data collection that required recounting the analysis, which turned out quite in line with the original results again, confirming the observed effect. That may be modest but real. Overall, treatment-induced reduction in coughing between 13 and 18% in these patients. Not all patients may perceive that as a tremendous improvement, and therefore it will be an individual decision whether that is a reasonable treatment for an individual patient or not. The regulatory decision for the marketing authorization, on the other hand, is clear. There is a positive CHMP opinion based on an admittedly modest effect confirmed in the 2 independent studies, with secondary endpoints supporting the primary endpoint and a benign safety profile. What do we learn from this case? If there's clear efficacy, here shown by 2 independent studies and supported by secondary endpoints, this efficacy can be sufficient for a positive opinion, even if the benefits may be rather modest. The next example, a marketing authorization application based on a single-arm trial, will be presented by Bruno.
Thank you, Sabine. And I bring you now the example of FINLY. Low and high-grade gliomas are rare pediatric tumor types in the pediatric population. Pediatric gliomas constitute approximately 46% of primary brain and other central nervous system tumors in children and adolescents aged 0 to 19 years old. FINLY is precisely intended for the treatment of low and high-grade glioma, LGG and HGG for abbreviations, as you will see across the slides. With this application, the applicant aimed for an indication for dabrafenib covering both low and high-grade glioma in combination with trametinib for the treatment of pediatric patients aged 1 year and older with BRAF V600E mutation, while for low-grade glioma, the indication is for patients who require systemic therapy. For high-grade glioma, dabrafenib should be given to patients who have received at least 1 prior radiation and/or chemotherapy treatment. It's acknowledged that available treatment options for both high and low-grade glioma are not satisfactory, and that a clear unmet medical need exists. FINLY received an orphan designation in 2020. With this treatment, an increased overall response rate and increased progression-free survival are expected, and altogether with the duration of response, are anticipated to translate into an important clinical benefit. The most commonly observed BRAF mutation is V600E, which has been identified in 17% of pediatric low-grade glioma and approximately 6% of the pediatric high-grade glioma. Oncogenic mutations in BRAF lead to constitutive activation of the RAS-RAF-MAC-ERK pathway, and dabrafenib is an orally available inhibitor precisely of the RAF kinases in this pathway. Dabrafenib will be administered in combination with trametinib, and this combination results in the inhibition of 2 sequential kinases in the MAP kinase pathway, enhancing this MAP pathway inhibition. The main study in support of this application combined 2 pediatric glioma cohorts into a single multicenter, open-label, phase 2 study. Patients were enrolled at 58 centers across 20 countries. The 2 cohorts consisted of pediatric patients with BRAF V600E mutation positive gliomas with either low-grade glioma, the LGG cohort, or relapsed or refractory high-grade glioma, the HGG cohort. The clinical relevance of efficacy in pediatric patients with BRAF mutated low-grade glioma treated with dabrafenib plus trametinib in first-line therapy is supported by sufficiently robust overall response data in this slide, and it's also supported by a substantial progression-free survival benefit, as reported from the randomized low-grade glioma cohort in study G2201. Considering overall survival, data are very immature to reach a conclusion. In addition, a clinical relevant overall response rate and a clinically meaningful response duration, in the next slide you will see it, is reported after treatment with dabrafenib plus trametinib in pediatric patients with relapse or refractory BRAF mutated high-grade glioma. Next slide, please. Despite the single-arm study design and limited sample size, activity is evident and can be isolated, which is deemed relevant in this scenario where there are no satisfactory treatment options. As recommendation post-approval, the CHMP then expects the final data delivery from the pivotal study, LGG and HGG cohorts, to be submitted. Due to early data cutoff, the applicant was also requested to provide updated survival data when available. Next slide. So in conclusion, in subjects with relapsed or refractory BRAF V600E mutated high-grade glioma, the demonstration of efficacy and safety was based on a single-arm trial as described. In contrast to PFS and overall survival, the overall response rate observed in a single-arm trial can to a great extent be ascribed to the experimental agent since spontaneous regression is not anticipated. Consequently, overall response rate must form the basis of the inference of a potential clinical benefit from the presented single-arm trial. Due to the limitations of overall response rate, the results should be outstanding in relation to what can be achieved with existing therapeutic options. Considering the poor prognosis of these pediatric patients with glioma and the poor outcome with current available treatment, the overall response rate at 46.6% for the low-grade glioma cohort and 56.1% for the high-grade glioma cohort after dabrafenib and trametinib combination treatment can be considered outstanding. Thus, the activity in terms of overall response rate, along with the duration of response, is anticipated to translate into an important clinical benefit as hypothesized. The lack of a randomized control arm in the high-grade glioma, by which we could isolate the effects on progression-free survival and overall survival, is acceptable given the level of activity seen and the lack of satisfactory treatment options. Overall, the benefit-risk balance of Finley in combination with trametinib was considered to be positive, subject to the conditions stated in the EPAR, and the CHMP wants to see further data as soon as it is available, confirmatory data. The indication as applied by the applicant received a positive opinion from CHMP, who recommended to the European Commission granting a marketing authorization to Finley. Back to Harold.
We have seen now positive examples that were sufficient for a positive opinion. One case where outstanding results in a single-arm trial were convincing, and one case where moderate efficacy was confirmed in 2 studies and supported by secondary endpoints. Next, we will have an example of a randomized control trial that failed to convince CHMP. Sohonos was intended to offer a therapeutic option for the treatment of fibrodysplasia ossificans progressiva, an extremely rare but horrible disease. There is no effective treatment, and the abnormal bone formation will slowly kill the patients. With other words, there is a tremendous unmet medical need. Sohonos, palovarotene, is a retinoic acid receptor gamma agonist and was expected to decrease the abnormal bone formation. The pre-specified endpoint in the clinical study was a volume of newly formed bone, quantified by whole body CT scan. Further pre-specified was that negative values would be zeroed, as there was no reason to expect any decrease in heterotopic bone. In addition, a square root transformation of the values for the volume of newly formed heterotopic bone would be used to decrease the impact of outliers. Both considered acceptable, reasonable when they were suggested. Sadly, however, the results of this pre-specified endpoint failed to show any effect. A p-value of 0.6 in the Wilcoxon test needs no further comment, I'm afraid. Only after this disappointing result was seen, an alternative evaluation was made, this time including the negative values, that is, this seemingly reduced volume of heterotopic bone, and this time without using the square root transformation for the limiting of the impact of extreme outliers. And now, in this post hoc analysis, there seemed to be a very clear treatment effect with an outstanding statistical significance. Looking then at the data for individual patients, it became very clear why extreme outliers and negative values for heterotopic bone formation make such a difference. There are more apparent outliers in the untreated group. There are more negative values in the treatment group. And while it would be fantastic if there was a way to decrease, to eliminate the heterotopic bone after it had been formed, there is no evidence that a retinoic acid receptor gamma agonist could achieve that. So, as a consequence, CHMP could not accept this post hoc analysis as a basis for the conclusion on efficacy. CHMP could not accept that this post hoc analysis could overrule the negative result with a pre-specified endpoint. So, in the end, there was a negative opinion. Efficacy results even for a conditional marketing authorization, must show some robustness. And here we have completely different results depending on how you evaluate the data. Clearly negative in the pre-specified evaluation. Post hoc modifications in the evaluation always raise the question if they might be data-driven, and they are difficult to use as a basis for a positive opinion.
Thank you very much, Harold. And let's go to Omblastis. So neuroblastoma, and I bring you back to another pediatric rare cancer. Neuroblastoma is a rare pediatric cancer that is the most common extracranial solid tumor cancer in children. High-risk neuroblastoma is a life-threatening disease associated with poor long-term survival. For high-risk patients, 5-year overall survival rates have improved during the last 2 decades, but are still below 50%. Omblastis came to us with a proposed indication for treatment of central nervous system and leptomeningeal metastases of neuroblastoma in patients who have previously received CNS-directed multimodal therapy for their disease, such as chemotherapy, surgery, or radiation therapy. Iodinated omburtamab recognizes and binds selectively to the B7H3 antigen expressed in the central nervous system leptomeningeal neuroblastoma tumor cells. This iodine radiolabel emits beta radiation, resulting in DNA damage and tumor cell death. The submission is based or was based in 2 trials. For convenience, I will call the 03133 just 133. That was a single center basket trial in the US intended to investigate dose finding and the toxicity of intrathecal administration of iodinated omburtamab. The majority of patients had neuroblastoma with leptomeningeal parenchymal metastases, and the trial was initiated in 2004, and data were recorded over more than a decade. Enrollment was presumably stopped in 2018. This trial provided most of the efficacy data with respect to overall survival, including long-term outcomes. But there's a second trial, 101 study, that is a multi-center single-arm trial that is still ongoing. This trial recruits only neuroblastoma patients with leptomeningeal CNS metastases. An interim analysis was conducted on data from patients recruited up to the 1st of January 2020. Further data were submitted during the procedure, and at that time, 50 patients had already been recruited, instead of the 32 planned. The most important favorable effect to consider is the 3-year overall survival rate that is derived from trial 133. The 3-year overall survival rate from the Kaplan-Meier estimate for the full analysis set was 0.56 with a 95% confidence interval. The median survival is estimated as 50 months with a 95% confidence interval. Next slide. The 3-year survival rate is a clinical relevant endpoint, as it could indicate long-term survival in patients with a malignant disease such as neuroblastoma. 12 months CNS or leptomeningeal progression-free survival is an unusual endpoint, but could be accepted in these specific circumstances. Supportive information can be derived from the 12 months CNS or a leptomeningeal progression-free survival rate, and that is estimated to be 0.59 with a 95% confidence interval. Notwithstanding, the interpretation of overall survival and progression-free survival without a comparator group is just not possible. The original purpose of the pivotal study 133 was dose finding and safety of an experimental targeted radiotherapy. From a confirmatory perspective, the insufficient planning and fundamental flaws in the trial design, the questionable choice of endpoints for the chosen single-arm trial, the changes in the conduct, and the analysis while the trial was ongoing were obvious concerns for CHMP during the assessment. Next slide. So it is a particular challenge to establish with reasonable certainty that the external control population is comparable to the trial population. The use of subgroups from external controls to make the results comparable with the finalized study 133 raised serious issues on the reliability of the results and their clinical relevance. In order to further explore the data and enable the use of the collected time-related endpoint, a propensity score method was used among other ancillary analyses that you see listed in this slide. The employed advanced statistical methods did not allow the establishment of efficacy, as they could not address the fundamental uncertainties related to the selection and unmeasured confounding. In any case, even despite this major effort in the development of the model, the obtained results are not considered compelling. As a general principle, and you can see it in the red banner, external controls in single-arm trials are only considered supportive to further explore the derived efficacy in case efficacy was established solely based on the single-arm trial itself. This is not considered to be the case in this application. So again, in conclusion, considering the dismal prognosis of the investigated study population, the described uncertainties related to data collection and interpretation could have been accepted if the efficacy of the product was clearly demonstrated, which was not the case with the data provided. Taken together, more prospective and unified long-term data are needed to provide meaningful results regarding the characterization of a safety profile in the claim indication. Currently, the quality of the data submitted merely enables a descriptive data interpretation. A favorable effect for iodinated omburtamab was not demonstrated, and the safety profile cannot be sufficiently characterized for the reasons already stated. And the CHMP considered that the benefit-risk balance of Omblastis was unfavorable. This is therefore a good example of a failed single-arm study. In the grounds for refusal depicted in this slide, the CHMP noted all these concerns identified that do not allow to adequately establish efficacy, and also noted some quality aspects, namely the stability of the active substance once administered, that could pose serious concerns, precluding approval. And I handle it back to Sabine.
The second big topic in this webinar is the conditional marketing authorization. Let's first have a look at what a CMA is all about. A CMA is not a lifeline for applications that otherwise may not make it. A CMA is a kind of shortcut for those most promising, potentially game-changing, innovative medicines.
You see here, for your reference, the legal text, as written almost 20 years ago. But rather than getting lost in subtleties of legal language, allow me to use an analogy. Just as we have different regulatory ways leading to marketing authorization, we have different roads in real life. The destination in the end is the same. The ultimate goal is the marketing authorization and making the medicine available for patients. But there may be different ways to get there, the highway or the gravel road. And the gravel road might be shorter, but more challenging. Which road you choose, that may depend on what you got. With a two-wheel drive or a truck, you may want to take the long, smooth road. If you have a four-wheel drive, you may go and risk the shorter gravel road. The standard application for a marketing authorization is a long, smooth road. Think the one suitable for a two-wheel drive, the one with only two requirements: a positive benefit-risk balance and comprehensive data. That's why you see a truck here, you're gonna need a truckload of data. The conditional marketing authorization may be the shorter road, but the more challenging one. Think, you need a four-wheel drive because you have four requirements. Firstly, a positive benefit-risk balance. Secondly, comprehensive data are expected post-authorization in the more or less near future. Thirdly, there is an unmet medical need that the new medicine will fulfill. And fourthly, the immediate availability of the medicine outweighs the risks of the less than comprehensive data, meaning the outstanding benefit is worth an increased uncertainty. Bottom line, outstanding benefit as trade-off for limited data. This is why a conditional marketing authorization may well be based on early limited data, on single-arm trials with only historical controls, in oncology on response rates rather than on overall survival or progression-free survival, if each and every one of the four conditions are met.
Let's first look at a positive example where a CMA was allowed in early access and was later transformed to a full marketing authorization. You will remember this slide from our first webinar when Harald introduced this figure as illustration for the difference in magnitude of efficacy and for the size of the uncertainty. Products suitable for CMA will usually fall in the category with a potentially high, possibly best-in-class efficacy, but nevertheless with quite some remaining uncertainty that will need to be addressed after marketing authorization.
Here we got an example that nicely illustrates the typical features of a CMA, with both its advantages of patients' early access to the medicines, with its major disadvantages, the high uncertainty. In 2016, Tagrisso got a CMA for the treatment of non-small cell lung cancer based on an overall response rate of 66% and a duration of response of 12.5 months. Undoubtedly an impressive efficacy, but the early availability came at the price of overall limited data from a single-arm trial that is with a higher uncertainty, which, by the way, was too much uncertainty for the German payers. Reimbursement, or rather the price, was an issue and resulted in a withdrawal from the German market. But one of the 4 conditions is that the comprehensive data will be available after the marketing authorization. And for Tagrisso, the data from the single-arm trial, when completed by PFS data from an RCT results that confirmed the clear superiority to the standard of care. This was enough to switch to full marketing authorization 14 months after conditional marketing authorization, and in the end brought Tagrisso back for the German patients too. A textbook example for a positive development. First, an early CMA, and soon after, the more data and the full marketing authorization. Except, of course, the disadvantage that the higher uncertainty at the early phase of a CMA may not be accepted by the payers, and therefore patients may not benefit from the new medicine regardless of the valid European marketing authorization. Of course, the higher uncertainty in the conditional marketing authorization will not always end well. Conditional marketing authorization will need to be completed or confirmed with additional data. And while most CMA in the end will become a full marketing authorization, unavoidably there will be some that will not be able to provide these data. Some will fail to confirm the positive view that we had when we granted the CMA.
Products suitable for CMA will usually fall in the category with a potentially high, possibly best-in-class efficacy, but nevertheless with quite some remaining uncertainty that will need to be addressed after marketing authorization. Of course, the higher uncertainty in the CMA will not always end well. CMA will need to be completed and confirmed with additional data. And while most CMA in the end will become a full marketing authorization, unavoidably there will be some that will not be able to provide these data. And some will fail to confirm the positive view we had when we granted the CMA.
And this will not only mean that the medicine does not advance to a full marketing authorization, it must mean that the conditional marketing authorization is withdrawn or revoked. Let's look at an example for expectations not fulfilled. Here we have such an example. Olaratumumab was developed as an add-on therapy to doxorubicin for the treatment of soft tissue sarcoma. The basis for a conditional marketing authorization were promising results from a phase 2 study. The trial with 150 patients in 2 randomized groups looked promising. Here you see the Kaplan-Meier for the progression-free survival, and the data for the overall survival seemed even better, even when we consider that at the end of the trial there were very few patients left. The plan was to confirm the benefit, the efficacy, in a multicenter randomized double-blind phase 3 study with 460 patients in 2 groups. Unfortunately, the results were not what we hoped for. Looking at the overall survival of the ITT population, the group treated with a combination of olaratumumab and doxorubicin did not look any different from the group that received only doxorubicin. And even focusing on the subgroup with leiomyosarcoma did not help. So the conclusion was inescapable. The positive benefit-risk balance has not been confirmed. As a consequence, this conditional marketing authorization application did not make the transition to a full marketing authorization. Rather, the marketing authorization was withdrawn. The higher uncertainty of a CMA is just that, a higher uncertainty, which means the seemingly clear positive benefit risk may not be confirmed in the end.
Another example for a conditional marketing authorization that did not make it in the end was Adakveo. And this case will be now presented by Bruno again.
Thank you very much, Harold. So Adakveo actually contains crizanlizumab, a selective IgG2 kappa humanized monoclonal antibody that binds with high affinity to p-selectin and blocks the interaction with its ligands, including p-selectin glycoprotein ligand 1. It is indicated for the prevention of recurrent vaso-occlusive crisis, the VOCs that you'll see across the slides, in sickle cell disease patients aged 16 years and older. It can be given as an add-on therapy to hydroxyurea, hydroxycarbamide, or as monotherapy in patients for whom hydroxyurea or hydroxycarbamide are inappropriate or inadequate. The recommended dose of crizanlizumab was 5 milligrams per K administered over a period of 30 minutes by intravenous infusion at week 0, week 2, and every 4 weeks thereafter. Next slide. At the time of submission, the main study included 198 patients with sickle cell disease showed that Adakveo was effective at reducing the number of painful crises. In this study, patients given Adakveo had on average 1.6 crises per year, while patients given placebo had an average of 3 crises per year. The study also showed that Adakveo reduced the yearly numbers of crises by almost a third in patients already taking hydroxycarbamide, 2.4 versus 3.6. and by half in patients not taking hydroxycarbamide, 1 versus 2. Next slide. The Kaplan-Meier curve also showed at that time the same clear picture that CHMP was assessing. Next slide. Given the limited treatment options, a high unmet medical need in sickle cell disease patients, the debilitating nature of the disease, and even though the data package was not yet comprehensive, it was concluded that immediate availability of the drug would outweigh the risk inherent in the fact that additional data were still required. The benefit-risk balance of Adakveo was considered at that time favorable for the prevention of recurrent vaso-occlusive crisis in sickle cell disease patients aged 16 years and older, based on the available evidence at the time and subject to additional confirmatory efficacy and safety data to be generated in the context of a conditional marketing authorization. In order to generate this comprehensive data that CHMP still wanted to see, using Adakveo, the marketing authorization holder that now will have a marketing authorization was requested to submit the results of 2 studies by December 2025 as specific obligations, the SOBs you see in the slide. They were the results of the primary analysis of the phase 3 82301 study, the STAND study, of crizanlizumab with or without hydroxyurea or hydroxycarbamide in adolescents and adults with sickle cell disease patients with vaso-occlusive crisis. But also we wanted to see the final results of the phase 2 82202 study of crizanlizumab with or without hydroxyurea and hydroxycarbamide in these patients with vaso-occlusive crisis. Next slide. In December 2022, the first interpretable results of the STAND study were communicated to the European Medicines Agency by the marketing authorizations holder. The study showed now that Adakveo did not reduce the number of painful crises leading to a healthcare visit. Patients treated with Adakveo had on average 2.5 painful crises, with a subsequent healthcare visit over the first year of treatment compared with 2.3 crises in the placebo group. In addition, the average number of crises requiring a healthcare visit or treatment at home was 4.7 with Adakveo compared with 3.9 with placebo. The results showed that neither the primary nor the key secondary endpoint, that was the annualized rates of veno-occlusive crisis leading to healthcare visit or leading to healthcare visit and treated at home combined, with crizanlizumab were met. These primary results, showing a potential lack of efficacy, raised uncertainty as to whether the benefit of crizanlizumab still outweighed its risks in its authorized indication. So, as a conclusion, the European Commission triggered what we call an Article 20 review, which is under the Article 20 of Regulation 726 of 2004. And here the CHMP assessed these new concerns and the possible impact on the benefit-risk balance of Adakveo. The CHMP considered all available data, including all the clinical data of the STAND study, study 82202, and other clinical studies, considered the real-world data submitted by the marketing authorization holder, as well as the views from patients and healthcare professional representatives who attended the oral explanation by the marketing authorization holder to the CHMP. Having all this in mind and considering all this data, the committee noted that no benefit was observed from treatment with Adakveo in sickle cell disease patients aged 16 years and older. As a consequence, the CHMP concluded that Adakveo lacks therapeutic efficacy and that the benefit-risk balance of Adakveo is no longer favorable. On the 26th of May this year, EMA's Medicine Committee, the CHMP, then recommended revoking the marketing authorization of Adakveo.
In the examples we have seen so far, both in the negative and positive examples, a conditional marketing authorization had been granted in the first place, even if, in the case of the negative examples, it was withdrawn later on. Of course, not all early results can be considered sufficiently promising to justify a conditional marketing authorization. Where to draw the line? For regulatory decisions, consistency is very important. Therefore, previous decisions usually provide some orientation. Tepotinib, Tepmetko, may be a good example to show where, why, and how we set a limit. Apparently, both a first-line and a second-line indication were considered based on response rate data in non-small cell lung cancer, with FDA going for the first line in the United States and EMA for Europe granting only a second-line indication. Let's see if this could have been predictable by looking at previous CHMP decisions for targeted therapies for non-small cell lung cancer based on response rates. Here you see the response rates that were submitted in previous positive decisions, including the example for Tagrisso we had earlier today. Previous positive decisions were based on a range of 62% to 73% response rate in the indication of non-small cell lung cancer first line. Tepmetko now showed a rather constant response rate of around 50% in the first line treatment, obviously less than the previously approved products. So, CHMP did not consider this as fulfilling the unmet medical need in this first-line indication. But CHMP did grant a second-line indication. Let's see how the response rate of Tepmetko in the second line compares to the response rate of other targeted medicines that have a second line non-small cell lung cancer indication. In the second-line indication, we had a broader range of previous results, from 40 to 60% response rate. And here, Tepmetko, with a response rate of 40 to 50%, depending on how the MET-positive patients were selected, got a positive opinion. Another positive example would be Rybrevant, which got a positive opinion for a second-line indication based on a response rate of 43%. And another negative example would be Exkivity, which had a response rate of 28% in the second line and was withdrawn after CHMP signaled a rather critical view. Bottom line, as regulatory decisions try to be as consistent as possible with previous decisions, these previous decisions are a good and helpful orientation and allow, to a certain extent at least, the prediction of what might be acceptable and what might not be acceptable for a conditional marketing authorization. The third big topic is how we use subgroup results. Always a tricky issue, but becoming even more important since our colleagues at the FDA are putting more emphasis on inclusion and diversity in clinical trials. It's going to be important to distinguish what we do with the general subgroups based on general criteria such as sex, race, or age, and with subgroups based on a medicine-specific pharmacological targets or its binding site. Let's look at examples for the general subgroups. Here, we have a seemingly clear difference between males and females. Looks like a clear separation of the overall survival curves for males with nothing doing for females. But a closer look shows that the finding is not very consistent. If we look at the event-free survival, there is no difference between the efficacy in males and females. In addition, there is no pharmacological rationale why midostaurin should work only in males, not in females. This is why this is considered a chance finding. Similarly, there seems to be a gender or sex difference in the efficacy of Keytruda for the treatment of non-small cell lung cancer if tumors had a high PD-L1 expression. Here, both the overall survival and progression-free survival results seemed to show this difference. But again, there was no biological possibility for that, and several other trials looking at the checkpoint inhibitors with non-small cell lung cancer did not show a relevant sex difference. Therefore, again, this finding in the smaller subgroup for females was considered a chance finding. The opposite finding with regard to sex differences was observed for the therapy of migraine. In the significantly smaller subgroup of males, the medicine seemed to lack a therapeutic effect. Again, there was no pharmacological rationale for that. Other medicines with the same mechanism of action did not show that, and the subgroup was significantly smaller. Therefore, while the observation was described in the European Public Assessment Report, this was no reason for a restriction of the indication to females or for an explicit warning in the SMPC. Again, for the same product, there appeared to be a clear difference in efficacy depending on body weight. As the group were divided as above and below median body mass index, there could be no argument that one of the groups would be much smaller than the other. But as you see on the slide, even in the high body mass index group, the treatment still appears efficacious, although less so than in the low body mass index group. Again, no reason to have any restriction of this indication. It is sufficient to describe this in the EPA, in the European Public Assessment Report. Is this caution, the reluctance to base regulatory decisions on subgroups, something new? No, certainly not. The discussion on the use of subgroups goes way back into the last century. A particular nice example was the ISIS-2 trial, assessing the effects of streptokinase and aspirin in patients with myocardial infarction. With overall 17,000 patients included, the mere size of the subgroup should not have been prohibitive for meaningful results. And so, the editors of The Lancet insisted, overruling the reluctance of the leading statistician Peto, on an analysis of subgroups. The result is a timeless jewel in the crown of scientific publications. Dividing those 70,000 patients in 12 subgroups, according to the zodiac sign of the patients, gave interesting results. No treatment benefit for Geminis and Libras, whereas, for example, Capricorn apparently profited from the treatment. These examples were meant to illustrate that the high number of subgroups assessed in most applications are of course prone to chance findings and are usually not type error 1 controlled. Therefore, in the absence of a pharmacological or biological rationale, these differences are described but are rarely reliable enough to justify restrictions on the use of the medicine. So, in general, subgroup analysis must be taken with a grain of salt. Or, to express this more scientifically, hypothesis-generating results in subgroups may trigger further research. All these subgroups we usually assess have some probability to give at least a few seemingly clear results. Unless confirmed, unless these results form a consistent and meaningful pattern, this research should be considered as hypothesis generating.
Things may be different when the subgroups are defined by the pharmacological target of the medicines under investigation. Even when the subgroups based on the pharmacological target do not result in change, do not result in a restriction of the indication, this is always valuable information for the understanding of the medicine's pharmacology and, more importantly, for patients and their physicians when they make individual treatment decisions.
Here we have a harmless example. For Trodelvy, the overall result was clearly positive. But as this medicine is a targeted therapy, targeting trophoblast cell surface antigen 2, TROP2, it seemed obvious to wonder if the expression of this pharmacological target in the tumor would make a difference for the efficacy of the medicine. In this case, only during the assessment of the marketing authorization application, additional data were provided, subdividing the overall population of breast cancer patients in different subgroups, depending on the quantity of the trophoblast cell surface antigen 2, the target of Trodelvy. It is easy to see that there is a difference between patients with high and low expression. Unsurprisingly, the therapeutic effect is significantly higher in patients with high expression of the pharmacological target in their tumors than it is in patients with low expression. The good thing is that even in the patients with low expression, there is apparently efficacy. They do better than the control group. So while this information certainly is of interest, it is not a reason to restrict the indication. It will be left to the individual treatment decision to find the best option for the individual patient.
In the example we just saw, efficacy was different in subgroups defined by the pharmacological target. Nevertheless, as all subgroups had a benefit, this did not change the indication or restrict the target population. Next, we have an example where indeed subgroup results for efficacy led to a restriction of the indication. Opdualag is a combination of the checkpoint inhibitors nivolumab and relatlimab. And this combination is obviously efficacious for the treatment of melanoma, as seen here in the progression-free survival results for melanoma patients. However, if we look at subgroups defined by the expression of PD-L1, the pharmacological target of nivolumab, a difference between subgroups appears. Therefore, the European indication limits the use of Opdualag to patients with PD-L1 expression in tumor of less than 1%. By the way, different from the US. FDA granted an all-comer unrestricted indication. It may seem surprising that Opdualag works better in patients with tumors that express less of the pharmacological target. Usually, we see the opposite with checkpoint inhibitors. While we have no definite explanation for this observation, it is tempting to speculate about a kind of ceiling effect. As you see on this slide, when treated with nivolumab only, patients with PD-L1 expression of less than 1% do worse, have a worse clinical course than patients with an PD-L1 expression higher than 1%, just as expected. It may be that for these patients, adding another checkpoint inhibitor, adding relatlimab, can achieve an improvement, whereas for the patients with a higher PD-L1 expression, nivolumab alone is more efficacious already provides all the benefit checkpoint inhibitors can provide, and therefore relatlimab has no additional effect. This would also be in line with the observation for the combination of nivolumab and ipilimumab, for the treatment of melanoma in this case. Looking at the overall group of patients, adding ipilimumab to nivolumab increases efficacy. But this effect is obviously driven by the subgroup of patients with PD-L1 expression of less than 1%. In patients with an PD-L1 expression higher than 1%, additional treatment with ipilimumab does not seem to have an additional benefit. Here you may see a learning curve for regulators. Back then, the information on the apparent lack of additional benefit in patients with higher PD-L1 expression was provided in the indication wording, but without explicitly restricting the indication to patients with lower PD-L1 expression.
The examples we have shown you all represent final positions of CHMP in these procedures. This does not necessarily mean that this position was uncontroversial when it was developed. Nevertheless, once the decision has been taken, the position sets a standard for future decisions, sets a precedent that is an important orientation for future decisions. It is, of course, desirable that the future decisions are consistent, in other words, follow the direction set by previous decisions. If, for whatever reasons, they do not, the rationale for this change of position needs to be clearly outlined. Maybe the case that a seemingly similar situation is not that similar after all, because maybe we have additional knowledge that was not available previously, which changed the overall conclusion.
Now, allow me to summarize what we covered today. The first topic was efficacy. For most, albeit not for all procedures, efficacy is a crucial point. We have seen examples of limited or small efficacy that nevertheless could be consistently demonstrated in 2 state-of-the-art independent trials. And we have seen examples of outstanding efficacy in early limited trials. And both were used as basis for positive opinions. We had an example of why post-hoc changes in the primary endpoint are not very welcome. Secondly, we looked at conditional marketing authorizations that are intended for outstanding, groundbreaking new medicines and are not meant as a rescue operation for weak submissions. Examples for conditional marketing authorizations also showed their major disadvantage, the high uncertainty in several fields, for example, regarding reimbursement decisions or whether or not the conditional marketing authorization will lead to a full authorization or to a withdrawal of the authorization in the end. And finally and thirdly, some examples showed that different results in classical subgroups, such as sex or gender, race or ethnicity, body weight, should be interpreted with great caution, as the high number of subgroups opens the door to change findings. While subgroups defined by the molecular target of the treatment may deserve more attention.
And this is the end of this webinar and concludes the first series of webinars where I try to give you a general introduction or overview of how EMA is conducting the regulation of medicines in the centralized procedure.
In the second webinar series, we will then focus on oncology, and we hope you will be back for this. Thank you for watching, and
goodbye for now.